Professor Graham Ball. I'm Professor of Bioinformatics at Nottingham Trent University in the Van Gies Cancer Research Centre. I've spent the last 16 years developing techniques for using computers to identify biomarkers of cancer and other infectious diseases. What we do is we analyse data from the whole genome, from the genes of a set of patients, and we try and identify things which tell us, or genes which tell us about how that patient will respond to therapy, which group that patient falls into, and whether they will progress quickly or slowly with their disease. What is the risk for that patient? We also are identifying markers that potentially could be targets for therapy as well. So the microenvironment within a tumour, I think, is quite a complex thing. And within the whole tumorigenesis path, the whole path in which the tumour grows, there are many influences on that. So, for example, there could be mutations in the genetic material which result in the tumour outgrowing and evading other processes. That certain genes may not be repaired, if you like, which causes a mutation. That has one effect on the tumour microenvironment. It gets to a point then after a while where the tumour grows, and at that point it needs to have resources. It needs to have oxygen and things to grow more. So you end up with a centre of the tumour which is often very hypoxic. It has little oxygen. That would kill the tumour normally, unless it undergoes a process called angiogenesis, or lymphangiogenesis in some cases, in which it can start to bring oxygen and resources in the tumour to grow further. So that's another feature which defines that. But as soon as the tumour starts to engage with the blood vessels and the lymphatic system, then it starts to be prone to the immune system. It can be attacked by the immune system. So there will be certain molecular markers there at that point that might attract the attention, if you like, of the immune system. But there will be other things that help the cancer evade the immune system. So, for example, some types of immune cells can invade the tumour. They're called tumour-infiltrating lymphocytes. That can have also a big effect on the outcome for the patient. So it's important that we look at all these things and understand what are the molecular drivers for each of those processes. With respect to bioinformatics and its utilisation in biology, there's been a big revolution in biology since the sequencing of the human genome. Because we know the way the human genome is working to some extent now, we can map it. We can start to measure all of the genes that are expressed in the body, and from that all of the proteins. Now, that's really a wonderful thing, but there are around 30,000 genes that are expressed in the body. That's a huge amount of information, and the human mind can't cope with all that at one time. So what we do is we use computer methods to search through all of that data to answer particular clinical questions. We use a form of artificial intelligence called an artificial neural network, and we found that that approach works very well at finding the genes that associate with particular things of interest. So they could be whether a person has cancer or not, whether their cancer will respond to a particular therapy or not, whether their disease will progress quickly. And so the approaches we've used give us a list of, or smaller list of genes that are important in that context. Once we have that list, we can start to say, okay, well, what are the drivers of that system? Because we're more than just a list of genes. In everybody, there's a whole set of genes that are interacting with each other. We're all a very complex mixture of the interplay between different genes and proteins. So what we want to find in a given disease is, what is the thing at the center of that disease? What is the thing that we could target with a therapy, or the thing that we could measure to say that person has that disease? So we use another form of a computer approach to look at how things interact, to model how things interact, and from that tell us what are the drivers of that disease state. And the early indications are that we can find drivers that when we target them, when we knock them out, we can change the whole phenotype of the biological system. So it's exciting, and this is we believe a new way forward for analysing such data.